An effective and proven way to combating laurel wilt infection in the avocado grove is continuous grove sanitation. Joshua Konkol, biological scientist at the UF-IFAS Tropical Research and Education Center, says such grove sanitation should be put in place as a first step. He advocates this is important even if the tree is just wilting.
And usually when a grower begins to see wilt symptoms, the tree is already beyond saving.
Konkol says ambrosia beetles infected with the laurel wilt pathogen seek out trees that are stressed, for example, by persistently wet soil conditions or drought, or by root rot pathogens. He says trees that are stressed produce ethanol, that beetles detect this and attack the tree.
The beetles will begin to fly to those trees, and they will begin to bore into them, and then they will carry the pathogen into the tree.
Konkol simulates that process by using a drill and a syringe to inoculate small container-grown avocado trees in greenhouse studies, spreading disease using ambrosia beetle-like delivery, artificial inoculation with the pathogen. The goal is adapting a similar strategy for use with inoculants formulated to control spread of laurel wilt.
So it's very important that we do both greenhouse and field experiments. So the greenhouse experiments are the small experiments that we begin to set up the field experiments, because the field experiments are sort of like the actual trial.
Konkol says inoculations are almost always done on trees that are being studied, those in the grove and those in the greenhouse. That way, he says, he has positive control, ensuring that experimental trees have been infected. So, inoculation first introduces the disease. With an electric drill and a syringe, the ambrosia beetle's vectoring is mimicked.
For the inoculation, we would take a 7/64-inch drill bit, and that's the size of approximately the size that ambrosia beetles create when they bore into trees. So we try to simulate an ambrosia beetle boring into the tree. We take the drill bit and we drill 4 holes on 4 different sides of the plant. And this is because sometimes you can drill into one side of a plant, and you'll drill into an area where the vascular tissue is damaged or something like that, and the pathogen may not move. And so we had some complications earlier on where we found that you would introduce the pathogen into a tree, but nothing would happen. Because if the xylem tissue is dead or compromised somehow, the pathogen can't move from that point.
After drilling 4 holes, inoculation begins.
We pipette the inoculum into the tree. Currently, we inoculate the tree with 3,000 colony-forming units, or conidia, or cellular components. We use colony-forming units because they're not really spores all the time. It could be cells. It could be a variety of different things that enable the pathogen to grow. So any part of the pathogen that is able to grow into a separate colony is what we refer to as a colony-forming unit.
It's then, by inoculation, that fungicides are tested for effectiveness at reining in laurel wilt. Over time, physiological differences within the tree will be recorded as infection spreads or does not spread. Konkol expects a recent Trek discovery to be helpful in controlling Rafflesia loricola. He says a separate but closely related Rafflesia species is not pathogenic, and that he expects comparisons of the 2 species may decrease the time needed to develop control strategies.
If you can begin to determine what genes Rafflesia loricola has that the other species does not, you can begin to determine what are the genetic components of Rafflesia loricola that enable it to become a pathogen.
Genetic differences between the pathogenic and the non-pathogenic species are now under study. This research, being conducted at the molecular level, is being done on UF's main campus in Gainesville by plant pathologist Dr. Jeff Rollins.
If we could study the pathogen at the molecular level, we really could try to understand those factors, those molecules that the pathogen is using to cause this disease that kills the trees.
In our next segment, Dynamics of the Pathogen, Rollins provides a deeper insight into his research of the determining factors that make up the pathogen, research which could provide infected avocado groves a second lease on life.